Thank you, everyone. Firstly, I'd like to thank ARM for hosting this very insightful workshop and for also having us, giving the opportunity for FormBio to present our work and get some ideas too. So we're very excited to showcase how we can utilize AI/ML models to improve the vector design for gene therapy. I know this audience knows very well that gene therapies help to address some serious medical conditions that have few or no alternative treatments. However, some of the biggest bottlenecks that therapy faces is design, safety, as well as manufacturing. So when we think about the design hurdles, we're thinking about the fact that the entire therapeutic payload that we intend to deliver needs to be packaged efficiently. If we have partially filled capsids, then that's going to cause a lot of off-target effects and compromise the therapeutic outcomes. And then it needs to be delivered to the tissue that we intended to. It needs to express, and it also needs to make functional protein. When we think about safety concerns, we want to ensure that we minimize the immunotoxicity, no long-term safety events. And when it comes to manufacturing, some of the hurdles are the scalability, ensuring that it's cost efficient, and also maintaining quality control. So when we think about some of the design challenges that we face during gene therapy, and when we address some of these issues, typically we note that, in the earlier stages, the discovery stages, we think about efficacy. That's where the main focus is. And as we move towards the later stages of IND and clinical development, we think about how we can scale this up. And that's where the manufacturability comes into play. And in the design stages, there are a lot of considerations that we need to keep in mind, which includes the fact that the therapeutic payload needs to be efficiently packaged. It needs to express in the right tissues. We need to ensure that there are no CPG islands that are present in non-promoter regions, because that could lead to immunotoxicity. It needs to have a good yield. So a lot of these factors have to be considered when we're designing these vectors. And the question then becomes, can AI/ML models help to address some of these concerns that we face so that we can also reduce the bottleneck of manufacturability at the later stages? So the question is, can we address manufacturability at an earlier stage in the discovery phase so that we can, it doesn't become as much of a bottleneck as we move towards the later stages of development? And so the answer is yes, we can use AI/ML models in silico assessments of encapsulated viral genome identity, immunogenicity, as well as manufacturability can help to de-risk vector design sequences and also optimize them so that we are coming up with vectors that are safer, more manufacturable, and have a higher probability of success. And so with that, I want to spend some time talking about the AI/ML model that we've developed in order for us to make these vector designs, or generate vector designs that have a lower truncation risk, thereby leading to more efficient packaging and more efficient manufacturability. So our foundational model is a large language model that has been pre-trained on nucleotide sequences, including from the NCBI, GenBank, the Human Protein Atlas, some patented viral sequences, published sequences, as well as proprietary data set. So we have invested heavily in our training data sets, because obviously that's what is important in order for us to generate outputs that actually provide us with insights. So as I mentioned earlier, the generalized training data set has been trained on nucleotide sequences from publicly available data sets. And then we also have task-based data, wherein we have sequenced hundreds of AAV gene therapy vectors using long-read sequencing so that we are able to make task-based predictions. So upon our extensible base model, we have primary tasks and subtasks that help to provide vector design outcomes and optimizations. So the idea being that if you input a nucleotide sequence into our model, we're able to generate these insights. So let me see if this works. Okay. So one of the outputs that we get is what we call construct truncation, or the truncation risk. So you can imagine this to be your vector sequence, and it's annotated according to the different regions. I'll explain this in more detail, but basically this tells us like which regions in the construct are more prone to truncations, and therefore you're not able to package the full intended payload that you want to, right? We also look at CPG islands and GC content because these could provide us or flag us with potential regions that can cause immunotoxicity when we inject this into mice or NHP. And we're also able to look at structural motifs that could cause truncations. So I want to spend some time now talking about some of the key challenges that we face in vector design and how in silico solutions can help address these challenges. So in the subsequent slides, you'll see some of the solutions that in silico assessments can provide. So the first challenge that we face is ensuring that the vector design efficiently packages the therapeutic gene of interest into the viral capsid, since this is very critical for a successful gene therapy delivery. Partially filled capsids can reduce the transduction efficiency and also compromise the therapeutic outcomes. Secondly, the structural motifs also can impact the vector's ability to be replicated and packaged efficiently into viral particles. Thirdly, we want to minimize the risk of immunogenicity. We do not want any adverse events because that can also compromise the therapeutic outcomes. Fourthly, we also want to optimize the coding sequence so that we minimize the risk of any premature truncation or termination because we want to maintain the functional integrity of the protein and enhance the therapeutic efficacy. And finally, we want to accelerate the screening of identifying the lead gene therapy candidate vectors so that we are expediting the development and also the translation of promising candidates into the clinic. So as I mentioned, one of the outputs that we get from our AI/ML model when we input a nucleotide sequence for AAV gene therapy constructs is it allows us to identify regions within the construct that are at a higher truncation risk. So did I forget that already? Right. So what we have here on the y-axis is the truncation risk that is plotted on the natural log scale. And on the x-axis, we have the different vector elements. So we can see that here's the ITR, the CAG promoter, GFP CDS, poly A tail, and then the right ITR. So we have the forward and the reverse replication sequence here. And when you see these peaks, right, in the promoter region, these peaks indicate that these are regions that are at a higher risk for truncation. So these are regions that are problematic, and that's something that's information that you get right away. So what can we do instead is that we can maybe swap it out for a promoter that has a lower truncation risk so that we're still able to package the therapeutic gene of interest most efficiently. Another output that we get from our AI/ML model is what we refer to as secondary and tertiary structure motifs. So on the y-axis, it is the likelihood that a given set of nucleotides will fold into a secondary or a tertiary structure motif. And on the x-axis, we again have the different vector elements. So when we look at the promoter region here, we see that there are all of these different peaks, right, at high magnitudes, indicating that there are a lot of these structural motifs being formed. And the ones in the shaded region are those that also correlate with the predicted truncation event. So we can flag these motifs that are shaded as problematic because you can imagine that when replication is happening and it's coming across these motifs, it is actually causing maybe replication fork stalling and it's falling off. And so we are not able to replicate the entire, it's not replicating through the entire payload that we intend to. And that's also going to cause partially filled capsids. And that's what we want to reduce. So that's another output that we get from using our AI/ML models. And then we also look at GC content and CPG islands. So when we look at the left y-axis, that's where we have the GC content that's indicated by the blue line. And here we're looking for regions in the construct where the GC content is above the biological norm of 50 to 55%. Because if it is above that, what happens is that it can lead to long-range interactions. It can lead to structural CPG islands, which all could be problematic. And on the right y-axis, we have the CPG islands. So here we are looking for CG dinucleotides, and anything that's above 100% is shaded. So that means that these are motifs that are at a high risk for being methylated. And these are also regions that can be triggered by the innate immune response pathway, usually via TLR9 signaling. So when we look at these motifs in the promoter, we tend to ignore because they could be very important in terms of gene expression. But when we see them in other parts of the construct, specifically in the CDS, they could be problematic because they could trigger the TLR9 signaling pathway. So all of these are insights that you're able to get very quickly when you're evaluating gene therapy vector designs. And so in this way, we can help you de-risk your vector design, provide recommendations on what would work well, how we can further optimize it so that we can reduce the truncation risk. Another output that we have is optimization of the coding sequence. And in this case, we're trying to reduce the truncation risk of the coding sequence. So what we have here on the y-axis is the truncation risk, very similar to the first set of graphs that I explained. And on the x-axis, we have the different vector elements again. So what we have here in purple is actually the initial construct, and pink is the optimized construct. And you can see that there is a difference in the truncation risk between the initial and the optimized, right? So the optimized construct is actually closer to the noise flow that's indicated by the black dashed line, indicating that optimization of the coding sequence is actually reducing the truncation risk. And so in this way, by optimizing the coding sequence, we are able to generate a library of new constructs with improved manufacturability and safety. So by, if you have, let's say, 20 or 30 or even more, like a large number of constructs that you want to evaluate for to identify what are the candidates that are expected to work best, we can run through all of those constructs through our AI/ML model and then rank the best candidates that are expected to have the least truncation predictions so that you know that these are the candidates that are expected to package well, that are expected to be safer with lower immunogenicity as well. And so this way, we're able to accelerate the development of drug substance. So coming to what is the biggest value proposition of using AI/ML models specifically for designing gene therapy vectors, it is that it helps us identify the vector designs that have the best outcomes. And so this reduces the trial and error cycles and also helps to accelerate the development of drug substance. So in silico assessments of vector designs can help us to provide actionable guidance on whether the design can be further improved or if the design is already very good, and how quickly this information can be available to customers. And so these AI/ML models allow cell and gene therapy developers move from preclinical development to IND faster, resulting in drug candidate products that are safer with lower truncation risk and fewer lab iterations. And so in closing, I obviously want to acknowledge everyone at FormBio for the amazing amount of work that's gone into making this happen. I also want to express my sincere gratitude to the Alliance for Regenerative Medicine for hosting this insightful workshop. Your commitment to driving progress and fostering collaboration within our industry is truly commendable. And on behalf of FormBio, I also want to express our sincere gratitude for allowing us or providing us with this opportunity to contribute as speakers and engage with fellow professionals. And so what we have here is a QR code that if you scan, you can see some of the latest experiments we conducted to test our AI/ML model outputs to see how well that compares with real wet lab data. So please check that out. I couldn't go through that in a lot of detail just because I wanted to make sure that I finish in time. And with that, we also had a couple of questions that we want to pose to the FDA, which are specifically about if there are any guidances on the use of master files, for computational tools, specifically for AI, the opportunities or advantages for having a CAT meeting, and how can the FDA actually facilitate collaboration between industry stakeholders, knowledge sharing between developers, so that we can leverage AI technologies efficiently for gene therapy vector design. And I'm sure you're going to hear this time and again that, for a good AI/ML model, we need good data. And so that is something that we need to address to see how we can, get the best data to train our models so that we can get accurate predictions. And lastly, how does the FDA take into account or interface with public and private consortia? And so with that, I'd like to thank you all for your time and open to any questions.
Yeah, thank you. Nice talk. I'm actually curious on your comments on the immune responses, because generally the GOI is the less immunogenic part of the gene therapy. And a lot of times it gets caught up in the immune response because of the amount of vector that we're dosing.
Yeah.
So have you been able to show that the de-risking actually results in a lower immune response via humoral and adaptive, or is that something you're still working on?
That's a great question. Yes, that's something that we're still working on. So we're working with our customers as they take our design vectors through in vivo studies to see how the safety profile looks like. But what we've seen very often is that some of the designs that we get from our customers, or some of the requests that we get from our customers, is to reduce the CPG island formation. Because what they have seen is that if they do have these CPG island peaks, they are seeing immunotoxicity when they administer that, in mice or NHPs. So that's something that we've often noticed that, hey, could you ensure that, you can modulate the CPG island so that it's not as high? And so we think that, some customers seem to care a lot about it and some don't. But that is something that it's a knob that we can tune in our model to see if, that is helpful for the customer or not based on what their needs are.
Right. I mean, the other caveat there is that the GOI you're putting into animals or for humans.
Yeah.
So they should be xenogenic anyway to some degree. It just depends on which residues are ultimately presented.
Yeah.
Which is kind of a conundrum why a lot of people sometimes don't care, or they're not as concerned about it.
Yeah.
The other question I had was around, the increased productivity. How are you measuring that? Because there's a lot of different ways to look at productivity and functionality in AAV.
That's a great question. So with the experiments that we performed, we were able to see that when we optimize the coding sequence, we're getting a lot more full reads because we are checking all of this through long read sequencing, because that's how we have trained our model. So we're seeing that there's increased full reads. About 28% increase is what we saw in one of the experiments we performed. And while this is not something that we control for, we also noticed that we're seeing an increased titer when we optimize the coding sequence. So that's how we are able to see the outputs of the optimization.
Yeah, I mean, just as a comment, it would be nice to see something like, and this would fit perfectly, Mike, with the workshop we did last year, but multi-probe DPCR, because I mean, a lot of IEX chromatography is going to grab anything with a charge on it from a genetic material, but doesn't tell you whether or not it's actually functional.
True.
And that's going to be the key question, I guess.
Yeah.
Thank you.
Yeah. Thank you.